So on the board here, I have drawn an artery and a vein, and I've got the wall, the tubular wall, layers in here. And so now we're going to continue on from our cross-sectional view to a longitudinal view. And so we're looking just like cutting it in half like a hot dog bun. And so basically we're going to label the layers of the wall. And so the first layer that I did was tunica externa, which is right here, tunica externa, okay, EXT. This used to be tunica serosa plus tunica adventitia, okay? So it's the outside layer right here, going all the way down, tunica externa. And so over here, it's the same thing. And so tunica externa. On the vein side also. All right, so now we're gonna do the next layer, which is the muscle layer. And so right here is gonna be tunica media. It used to be tunica muscularis, right there. And so over here on the artery side, right here, tunica media. Used to be tunica muscularis. And this is the layer that determines the size of the blood vessel by counting the number of muscle layers in it. So here, this one, we count muscle layers, and that's going to equal the size of the BV, okay? So right here. All right, sizes are determined by the number of muscle layers. So the tunica media. So tunica media is all the big layer of green. We have it over there. And so now let's count muscle layers. So to be considered a large artery or large vein, you have to have 10 layers of muscle. 10 layers of muscle in the wall, okay? 10 plus layers. 10 plus layers right there to be large, okay? Right, to be medium size artery or vein, well, we only need 5 to 9 layers. 5 to 9 layers of muscle. And then to be small, 3 to 4, okay? 3 to 4 makes a small size. And then extra small, 1 to 2. 1 to 2. And a capillary has none, 0, no muscle. So a capillary is a 0 for muscle, right? All right, so now let's put the numbers up on top on the vein side. So an extra small vein will be 1 to 2, small 3 to 4, medium 5 to 9, and large is 10 plus layers of muscle, okay? All right, so that's nice. And so now we're gonna label the last tunic because remember there's only 3. And so the inside tunic right here, the inside tunic, and I'm going to circle it right here because it's a big one, right here. This one's going to be called tunica interna. Tunica interna, and it used to be tunica submucosa and tunica mucosa. Still has those layers, but they're still hard to see when you see it on a slide, right? And so then we have the inside layer of simple squamous ET right here. Put a star on it, the star right here. And so the star is the endothelium. Endothelium right here. And so it's the inside simple squamous layer, right? So S, SQ, ET. And the simple squamous goes all the way down here, and then it becomes the capillary. So the capillary is made out of simple squamous ET cells. And then the simple squamous continues, and it's the inside layer, inside your veins, right there. Now, one other thing is the simple squamous has to be fed. You have to bring in food via the capillaries. And so right underneath these simple squamous ET cells is a little bitty layer of loose CT cells, loose CTs and capillaries. And so this is the lamina propria, and they will feed those little simple squamous ET cells. And then the green I have right here is basement membrane. Remember, all simple ET has to have basement membrane underneath it for support and diffusion purposes also. All right, so anyway, that's the basement membrane in green with a little bit of loose CT in the capillaries right there to feed the simple squamous ET cells to keep them alive, right? Right, and then in the lumen, of course, in our lumen right here, our lumen, we have plasma, all right? So this is our lumen, and so going all the way across our lumen, okay, our lumen, and then we have valves in the lumen. And so the valves are tunica interna, and I put arrows here. So in veins, again, every size of vein is going to have a valve. So that blood is going to push it up, and then flaps back down, and then that prevents backflow. So every size of vein is going to have a good old valve made out of tunica interna. The only one over here on the artery side that can have a valve is the arteriole, the extra small artery, and there it is right there. And so it will have a little valve to prevent backflow from the capillary. So it forces the blood forward into the capillary, right? So that would be it. And then the only other valve in an artery is at the beginning of the aorta, when the aorta starts coming out of the heart. And so the aorta is not part of the heart, it's a separate blood vessel. All right, so basically, we already know a lot of information about capillaries, and so we've already done this at the beginning of the semester. Basically, a capillary is either fenestrated or non-fenestrated. And what does that mean? If it's fenestrated, it's a fast delivery method, and it's got little spaces between its simple squamous ET cells. Basically, that allows for oxygen, waste, food, hormones to slide right through and into your body cells, right, into the cells that it's being delivered to. And so if it's a non-fenestrated capillary, basically there are no spaces between the simple squamous and the food items, the hormones, the oxygen, the gases. They all have to go into the cell and then diffuse out of the cell through the basement membrane and then into another cell where it's being delivered to. So it's a slower delivery method. And remember, non-fens are mainly in where? Nerve tissue and the testes. Only the testes of the male repro system. The other organs have fenestrated, leaky, but only where the sperm are developing, we have non-fens, right? All right, so that's basically a little review of how we get the sizes for the blood vessels, the names of the walls. So basically you just look at this is like slicing it in half and looking in. And so our three tunics are right there. And then the specific layer of simple squamous is always called endothelium. So if you hear of endothelial type diseases or disorders, it usually has something to do with the inside layer of your blood vessels. And so that would conclude everything you need to know about BV structures. Good job. This podcast is going to be on blood vessels, part of the circulatory or cardiovascular system. And so when you say cardiovascular, vascular always pertains to blood vessels. So BVs is the official abbreviation for blood vessels. And we already know there's only two kinds of blood vessels, arteries and veins. Arteries will always have a red color associated with them for color coding purposes, and veins are always blue. Now, it is a true statement that you cannot say that all arteries carry oxygen. That is false. It depends upon what circuit the blood is traveling in. And that will be part of other lectures on the cardiovascular system, blood circuits. So arteries, the first thing we can say about every artery in the human body is it always takes blood away from an organ. It's going to take it away from an organ, like the heart. Essentially, it can take it away from an organ, and we have to qualify that. And so it's going to take it away and take it to another organ, right? So from the heart, so it takes it away from the heart, all right? And then on that statement, takes blood away from an organ, which means equals the heart. Then it takes it, arteries are going to take it into another organ, okay? Into another organ. And so then it goes into another organ. Into another organ. So that's the big statement we can make about arteries. Now, are there other things that you should know about arteries in general? Yes. First of all, when you're looking at them on slides, or just even grossly in a cat or a cadaver, they have a more rounded lumen. They're more rounded, and they tend to be smaller in overall size, smaller. And so more rounded lumen and smaller in size. Right. And so why is that? It's because down here, all arteries have elastic fibers in their wall. And so elastic fibers are the same type as collagen fibers, which are connective tissue protein structures. So elastic fibers give it the stretch, the flexibility, and so also holds it really tight like Spanx. And so elastic fibers are found in the wall of the artery. So this half of my drawing, I'm just going to put elastic fibers in here. And so you're going to see this on slides. You cannot see it grossly, but these will be the elastic fibers on the artery side of my drawing. So elastic fibers will look like this. Little wiggles, squiggles right here, elastic CT, ECT. All right, perfect. So basically, when you have a tighter wall, then basically you have good peristalsis, right? You got good push power, and so the blood moves very, very quickly in the arteries. All right, then, another true statement about arteries is it does have valves, but not as many as veins. Veins have valves in all sizes of veins, but in arteries, we only have valves in two places. We have it at the beginning of the aorta, called the aortic valve, and then we have them in your arterioles to prevent backflow, and that's especially important in the kidney so that we don't have the backflow into the glomerulus. And so there, the afferent and efferents definitely have those valves to prevent backflow into the glomerulus or basically back into another artery. So those are about the main things we can say about arteries, all arteries, okay? Now, before I label this picture of the wall of an artery, and we know it's a tubular organ, let's go over and compare veins to arteries, okay? So I wrote it in blue because veins are blue. And so, first of all, they have an irregular shaped lumen. Very strange looking lumen. It's not round, it usually is stretched out, irregular shape. Then, the next thing I can say about all veins is it takes blood into an organ, okay? Into an organ. And what organ does it take it into? Basically, the heart, right here. And so when you say veins take blood into an organ, you have to be talking about the heart, okay? So the IVC and the SVC are two huge veins that are going to take all the blood into the right side of the heart, all right? And then after that, after you talk about the heart, then what are veins going to do? Veins take blood away from an organ. Okay? Away from an organ. And that's a true statement. So it's just the opposite of our arteries on the other side, okay? And so when you're talking about the heart, it's different than the rest of the organs in the human body, okay? All right, then the next thing about veins is they have no elastic CT in their wall, none. And so basically they're gonna be stretched out more easily, and so they stretch out, and when the vein wall stretches out, so it's called varicose. And you've heard of varicose veins. Can arteries become varicose? Yes, they can, but it's more rare because basically they've got that elastic to fight back to hold their size and shape better. But anyway, they definitely, the veins get stretched because of no elastic fibers. And so because they have no elastic fibers, veins in general are larger and bigger, and they do carry more blood, actually. So that's because of the lack of the elastic fibers. And we also have valves in all sizes of veins, every size, the venule, the extra small, the medium, and the large. And so they prevent backflow, right? And so no backflow. And so that's the function of the valves. And so now I'm going to use this picture in the middle, okay? In the middle to basically illustrate the wall of a typical artery and vein. And so what I did, I used our typical colors for the wall here, and I divided half of it into the artery side and half into the vein side. Now you're going to like this because before we always had 5 tunics to deal with, but because the tunics are hard to see in blood vessel walls, basically we're going to only have 3 tunics, okay? 3 tunics. So I'm going to label these for you. So we're going to start with the outside right here. And this used to be tunica serosa right here, simple squamous, and the dense reg underneath. Okay, great. Tunica adventitia. Now, these 2 are going to get renamed into our new tunic. And so what do we call it? You're going to like this. Tunica externa. Very easy. External on the outside layer. And so tunica externa. So that's going to be those 2 combined. Great. Now we do tunica muscularis, which is always the biggest layer. And so basically right here, tunica muscularis. All right, we've got that. And now this is going to become something else. And so it's going to be called tunica media. Media means it's in the middle, okay? It's in the middle. Tunica media. So it's renamed. And then our last layer is going to be here. And so tunica, you had tunica submucosa, all right, which is the dense reg. And then we had all this, our 3 laminas right here, tunica mucosa. And now we're going to rename this to tunica interna. Tunica interna. And so it's the inside layer of all blood vessel walls, right? And now they even renamed the lamina epithelialis because now they have a specific name for it right here. So lamina epithelialis is equal to endothelium. Endothelium, it is still simple squamous ET, and that's what all your capillaries are made out of. And we'll do one more drawing on the sizes of blood vessels, and we'll use these tunic names in that drawing. All right, so basically, that's the wall. That's the wall. We had the 3 laminas before, lamina muscularis, lamina propria, the loose ET, and then we always have the lamina epithelialis, and now that simple squamous inside of all blood vessels is always simple squamous. All right, so now we have to draw a valve, okay? A valve. And a valve is essentially going to be made out of basically the tunica interna. Oh, one other thing about the tunica interna. Sometimes, like if you read the book, it is called the tunica intima. Intima. A lot of people don't use that anymore because it's intimate with the blood. Whatever is what I say. And so I like tunica interna because if you use externa, what's the opposite? Interna. So I would not use intima, but basically there are some doctors that do like to use that term. All right, so now let's make a valve, okay? And so a valve is made out of the same layers, whether it's in an arteriole over here or in the aorta, and it's going to be made out of the tunica interna. So the first thing we're going to do is we're going to make some simple squamous ET cells like this. And then they're going to come down, little thin ones, just go like this. So basically, it's going to have, this is going to be a valve. And it's going to be made out of tunica interna. And here we go. So here's the simple squamous right here. And then we have to put some loose CT underneath it. So the loose CT is going to come in here and feed it with capillaries, okay? So it will be feeding it, the loose CT. All right, so it's in there. And now I gotta add some muscle. And so right here, it used to be lamina muscularis is gonna come up in here. And so you're gonna have muscle that helps move this thing, like this, like that. And then we can add some submucosa. And so basically the submucosa is gonna come in here, right here. And so it goes in there in the middle. And so basically in a valve, the dense irregular CT becomes dense reg CT. It just stretches out, and so it gives it more support in the middle of the valve. And so basically this is a one-way valve. When the blood comes through, it'll push back, and then when the blood goes through, it flips back and it seals the lumen, right? And so everybody knows the lumen, right? And so big L, right, the lumen. So this structure is your valve, right here, your one-way valve, made out of the tunica interna, right. So that's the basic anatomy in a cross-sectional view, comparing an artery to a vein.